This is the lecture for Lab 9, Energy Production. This is an overall equation for cellular respiration, and this involves taking the reactants, glucose and oxygen, running them through a series of reactions in order to release energy. So the ATP is the main purpose of this process. But in addition to the ATP, there are the waste products of carbon dioxide and water are produced. Mitochondria are sometimes called the powerhouse of the cell because they are the site of respiration in eukaryotic cells. And this is a diagram of an animal cell, which is one of the types of eukaryotic cells. And if you notice, this animal cell has mitochondria in it. And the purpose of the mitochondria is to perform cellular respiration. And cellular respiration has 3 stages. It has first glycolysis, then the citric acid cycle, and then the electron transport chain. The overall reaction for cellular respiration is again glucose plus oxygen run through a series of reactions to produce ATP, will then release carbon dioxide and water as waste products. And the energy, the ATP, is adenosine triphosphate. And that is the readily usable energy for the cell. And the purpose of cellular respiration is to dismantle glucose, to break glucose down in a series of steps, to slowly reduce all the energy in the form of ATP. And during this process, oxidation is the loss of electrons, and the glucose will be oxidized, and reduction is the gain of electrons, and the oxygen will be reduced. The first stage of cellular respiration is glycolysis, and this occurs in the cytoplasm of the eukaryotic cell. And glucose starts this process. Glucose goes into the process, and 2 pyruvic acids exit. And during these reactions, ATP and NADH are produced. So this is a diagram of what is happening during glycolysis. It begins with glucose, and the glucose runs through a series of reactions, and eventually it is split into 2 pyruvate molecules or 2 pyruvic acid. And in the process, ATP is generated. So some ATP is generated, as is NADH, which is an electron carrier. Before the citric acid cycle, there is a bridge or prep reaction, and that's where the 2 pyruvic acid are converted into acetyl-CoA so they can enter into the mitochondria. And during this process, a couple molecules of NADH is produced, and remember those are electron carriers. So here again, the pyruvic acid, it will be converted into acetyl-CoA so it can continue on into the citric acid cycle. And during this process, NADH is produced. The next major stage in cellular respiration is the Kreb cycle or the citric acid cycle. And this occurs in the mitochondrial matrix. During this process, the 2 acetyl-CoA are going to enter the process, and they are going to be completely oxidized into carbon dioxide. And during this process, the energy is removed and used to produce ATP, NADH, and FADH2. And NADH and FADH2, those are electron carriers. And this is an example of that process. So the pyruvate, remember, it's converted into acetyl-CoA so it can enter into the mitochondria. It goes through the Kreb cycle, and in doing so, carbon dioxide is released as a waste product, ATP is produced, and the electron carriers, NADH and FADH2, are also produced. The last part of cellular respiration is the electron transport chain. And the electron transport chain is actually a physical structure that is located on the inner mitochondrial membrane. And this is where all those electron carriers that were generated during glycolysis, the bridge reaction, and the citric acid cycle, they are bringing their electrons to the electron transport chain. And this chain is made up of proteins, and the last protein is the ATP synthase, which will produce ATP. And this is the stage where most of the ATP is produced in cellular respiration. This is a diagram of an electron transport chain. And an electron transport chain is a series of proteins that are embedded in a membrane. And this is found inside the mitochondria. And this is where those electron carriers, the NADH and the FADH2, drop off the electron to these proteins, and then that electron is passed from protein to protein through these complexes. As those electrons are transported along the proteins, that flow of electrons powers these large proteins, which are proton pumps, and they will pump hydrogen ions, otherwise known as protons, out to one side of the membrane. And this will build up a high concentration of hydrogen ions on that side of the membrane, so that when it comes to this ATP synthase, the ATP synthase will allow those hydrogen ions to diffuse back across the membrane. So they're diffusing from an area of high concentration to an area of low concentration. And this flow of hydrogen ions is going to power the ATP synthase to produce ATP. And at the very end, this electron, so the electron, once it was dropped off by the electron carriers and transported along the electron transport chain, that electron is going to meet up with oxygen and hydrogen and form a molecule of water. And in cellular respiration, you can generate, through the whole process, you can generate a maximum of 38 ATP per 1 glucose molecule. As a review of cellular respiration, the first stage is glycolysis. Glycolysis is where the glucose is converted into 2-pyruvic acid, and in that process, ATP are generated and also some electron carriers, NADH. Then the pyruvic acid is converted into acetyl-CoA, so it can enter into the citric acid cycle. During the citric acid cycle, the acetyl-CoA is completely oxidized to form some ATP and also other electron carriers like NADH and FADH2, and the waste product is CO2, carbon dioxide. Then those electron carriers travel to the electron transport chain where they go through the process to generate the largest amount of ATP during this process. And again, from 1 glucose molecule, a maximum of 38 ATP can be produced. This process is also referred to as aerobic respiration because during the electron transport chain, oxygen serves as the final acceptor, and it combines with hydrogen and that electron to form water. So this is referred to as aerobic energy production because it involves oxygen at that last stage. When oxygen is not present in an environment, that environment is considered anaerobic. Ana means without, aero means oxygen. And it is still possible for some cells to produce energy even without oxygen. Fermentation is one of these processes. In the process of fermentation, no oxygen is used, so this can occur in anaerobic environments. And this is basically a type of glycolysis. So no Krebs cycle is involved and also no electron transport chain. The end products of different types of fermentation can be lactic acid or ethanol. And during this process, 1 glucose molecule will produce only 2 ATP molecules. There are 2 common types of fermentation. The first is alcoholic fermentation. Alcoholic fermentation, again, is a type of glucose. It's where the glucose is split into 2-pyruvic acid, and then that goes through a process to release carbon dioxide and produce alcohol. The second type of fermentation is lactate or lactic acid fermentation, and this is where the glucose goes through glycolysis, but this time it is converted into lactic acid and there is no carbon dioxide produced. In lab, you will be using fermentation tubes for some of the experiments. This is an example of a fermentation tube, and in the fermentation tube, you will mix a suspension of yeast. Those will be the cells that will be performing fermentation. You will also add carbohydrates as a source of glucose for the process and water. And that will create a solution, and you will tip the tube so that the solution will go all the way to the top of the tube. Then over time, as the yeast are fermenting the glucose in the solution, CO2 will be produced and it will gather into a bubble at the top of the tube. So this is an example of a bubble of carbon dioxide that was produced by yeast in this fermentation tube, and then you measure the height of the bubble, and that gives you a relative reading of how much fermentation occurred. In another experiment, you will be looking at respiration rate. And remember, the equation for respiration is glucose and oxygen is needed to produce energy, and the waste products are carbon dioxide and water. So in this case, when you're trying to determine if respiration is occurring or not, you can look at the reactants to see if they decrease over time, or you can look at the products to see if they increase over time, which would indicate that respiration is occurring. In experiment 2, you are going to be looking at the level of glucose and what happens. And in order to determine if glucose is present, you are going to use the Benedict's test. So the Benedict's test involves adding Benedict's reagent and boiling the solution for 3 minutes. After that 3 minutes, it will change color. Blue solutions have no glucose present. Red solutions have large amounts of glucose present. So by looking at the color of the tubes, you can get a general indication of the relative amounts of glucose that are present in the tube.